Okay, so I think we'll start the session for today. So I'm just sharing the screen. Okay, somebody let me know.
Sir, actually, sir, it's visible. However, I have a question regarding enrollment. So, asking for a friend that
Mhm.
she wants to enroll in this course.
Yeah.
So, is the last date over for enrollment?
Uh, the last date, I believe it should not be over yet. Let me just check the information on the screen. Okay. Oh, okay. So now here it's showing February 2 actually. February 2, 2026 is the enrollment end date.
Okay, sir.
But here, I think you can still join because I see an option for joining at the top of the course here. It's saying enrollment ends on February 2, 2026. So that should be today, right?
Okay,
Uh,
sir. So, I can, my friend can join after the
yeah,
class.
yeah, that's fine. That's fine. But yeah,
Okay, sir.
I'm actually able to join. So it is opening up that form where you have to fill up some details, right?
Okay, sir.
Yeah, the email ID, institute, and qualification, etc.
Yes,
So
sir. The
yeah.
for signing in for the SWAYAM
Yeah, so
portal, right?
yeah, so after you sign in
Yeah.
using your SWAYAM account, so you go to the NOC 26 BT34, that bioinformatics algorithms and applications page, and then there should be still a join button available. Uh,
Okay, sir.
yeah, but it says 2 February 2026 is the end date for enrollment. So make sure that they join today. Okay.
Okay, sir. Okay,
Yeah,
sir. Thank you so much, sir.
yeah, it took some time, but it's fine. So I was actually looking into one of my other screens. So that's why you couldn't see it. Okay. So now I believe the screen should be visible here. Is that correct? Is my slide visible? Very confirm. Okay, I
Yes,
see.
sir. It's visible.
Yeah, okay. Thanks a lot. Thanks a lot. Yeah, so again, I welcome all of you to week 2 live tutorial session for the course Bioinformatics Algorithms and Applications. So this course is taken by Professor Michael Gromiha from IIT Madras. And for this live session, so there will be 2 TAs. One of them is me. So I'm Sunanda Samanta briefly. I am the PhD scholar from IIT Indore working in computational biophysics. And what kind of we do in this live tutorial sessions is that we talk about some of the concepts that Professor Gromiha has gone over in that week's kind of lecture content, and then we try to talk about some of the example problems that you might be facing in your regular assignments, right? So yeah, so earlier also for week 1 we took a lecture. I think there was another lecture on Tuesday also. So in this Tuesday there will also be a lecture, uh, and we will both kind of cover basically broadly the similar topics with maybe different approaches, but uh, that's for you to join according to your convenient timing basically. So whichever timing you favor, you can join the TA session. You can join both of the TA sessions, and you'll also get link to the uh, videos, the uploaded videos on YouTube, where you can maybe rewatch or watch the content that was covered in the live TA session. Okay. So in week 2's content, what has Professor Gromiha gone over? Can somebody tell me? So I think from previous classes, there was also some problems that I wanted to go over, but I couldn't. So in this class, we will also talk about those kind of problems. In previous weeks lectures, there was also some talk regarding databases, right? Professor Gromiha had introduced the databases, varieties of databases, and so on. So I'll also briefly cover those as well. And in this week 2's lecture, anybody can remind me what has been covered by Professor Gromiha?
Sir, so yes, sir. So in week 2 lecture, a major portion was given on Uniport demo.
Okay.
Then we also in his first 2 lecture he talked about basic protein like peptide bond, amino acids, the code for amino acids and all. And then he talked about functions of proteins like RB protein
Mhm.
and membrane proteins and also. Then in his uh, then he talked about pairwise alignment of the protein
Okay.
and nucleic acid.
Yeah. So concepts regarding alignments, proteins, nucleic acids, and so on, right? Yeah. So we'll also see. We'll try to cover some of those as well here. So just to brief on some of you on what happened on the first week's lecture, we basically talked about the different parts of bioinformatics and how basically all of the core sciences like biology, physics, chemistry, IT, computing, statistics are coming together to form kind of this intersection core domain where we basically try to analyze and play around with biological data and information, right? So that's kind of the bioinformatics part. It's applicable in a lot of variety of biological problems ranging from your genome sequencing to your protein structure prediction to subcellular localization, alignment, and so on. Protein-protein interaction prediction, drug discovery, and also a big part of it was database construction and development of software, so that basically the others in the community, in the scientific community, they can also benefit from your work directly via some usage of software, web servers, or different databases, right? We talked about some of the significance of bioinformatics studies. We talked about 5 major basically parts, such as database, derivation and testing of hypotheses, web servers, virtual screening, and big data analysis. And that's how I kind of wrapped up the earlier class, along with which talked about a few questions here and there. So in this particular class, so earlier, we talked about how different sequence-based parameters. So sequence kind of analysis is a big part of bioinformatics, right? As a whole, and one of the major ways that the sequences can be used or has been used for bioinformatics part, of course, are sequence-based analysis or sequence-based parameters or derivation of those, right? So sequence-based parameters are basically, let's say you have a big chunk of sequence, right? Now, if you want to calculate some sort of thermodynamic parameter or some sort of rigidity, flexibility, or some other sort of parameter from that big, let's say, sequence, big length of sequence, or a big number of samples of sequence, so it would have been very convenient if you had earlier the data for basically small constituting parts of those sequences that you already had the particular parameter for, so that you can basically kind of average all of those up or sum all of those up in your throughout your entire length of the sequence that you want to calculate the parameter for, right? So that is kind of how you get a sequence based parameter. You have to have the constituent parameter, or you have to have the parameters calculated for certain constituent sequence parts, like short stretches of sequences, for example, dinucleotide or trinucleotides. And then from that, through that particular values, you can get the overall value, overall parameter value for a big stretch of sequence, right? So, of course, to do that, you first need to understand, so what are dinucleotide and trinucleotide? So these are basically 2 stretches of consecutive nucleotide sequences, right? Trinucleotide will be 3 stretches of, so 3 kind of units taken together at a time, that will be count as a trinucleotide sequence, right? So in this particular question, it is asking that if you have sort of this big sequence, uh, you know, in its entirety, so how many dinucleotide and trinucleotide sequences you can get from this particular one? Any idea? So the dinucleotide or trinucleotides are basically short stretches of sequences for 2 or 3 nucleic acids, nucleotides, one after another, right? So any idea how many can you get from this big uh, length of sequence? You can give the answer in the chat. We'll just look into it. So this is the overall state of the sequence. And in this case, to get the particular number of dinucleotides or trinucleotides, you first have to get an idea whether the nucleotide sequences that will be considered should be overlapping or non-overlapping. Okay. So when we analyze a lot of bioinformatics sequences, usually in this particular case, for example, in sequence-based parameters, we always consider overlapping sequences. Okay. So overlapping dinucleotides or overlapping trinucleotides. So think of it in that way. I see a lot of C answers in the chat. So basically here the number of uh, constituents in the sequence, that is n equals 13, right? So a total number of 13 nucleotides are there, okay? And if you want to get the overlapping sequence, you have to basically start from each single of the constituent and see for how many of those you can move forward, let's say for trinucleotide 2 more steps or trinucleotide 1 more step. So what I mean by that is, let's say you want to get, just see. Yeah, so if it's n equals to 13, so if you want to get the number of overlapping dinucleotides, basically trinucleotides first. So what you have to do is that you have to start from each point and see if you can move 2 more steps forward, because these 3 nucleotides together will give you a trinucleotide sequence, right? So if you start from A, then you have to move forward 2 more steps and you will get AGC as a trinucleotide sequence. Now the next trinucleotide sequence that we will consider is that will be starting from G, right? And then starting from G, again you move forward 2 steps, you get GCA, right? So AGC, GCA, these are your trinucleotide sequences, overlapping trinucleotide sequences. The next trinucleotide sequence, you will start from C and then it will move forward 2 more steps. You get CAT, right? So AGC, GCA, CAT, then ATG, TGT, so on and so forth, right? If you want to get dinucleotide sequences, you have to move only 1 step forwards. For example, if you start with A, so AG will be your first dinucleotide sequence, then GC will be your second, then CA will be your third, and so on. So there is a handy formula if you want to get the number of trinucleotides or dinucleotides. It is n minus 2 for trinucleotides. Why n minus 2? Because since you have to move forward 2 more steps to get a particular trinucleotide, uh, if you think of it in this way, for the last 2 nucleotides, right, you cannot move 2 steps forward because there is nothing in front of that, right? So for this A, you can only move 1 step forward, which will give you this the A at the last, right? And for this A, there is nothing forward, so you cannot move any step from here. So basically, what this means is that starting from every point, this particular length of sequence, except for the last 2, you can always get a trinucleotide sequence by moving 2 steps forward. So in this case, you can exclude those last 2, and that n minus 2 value will give you the total number of trinucleotides. So that is 11, of course. And then for trinucleotides, again, since you have to see from how many points you can move 1 step forward, so that will be excluding this last A only. So right, so that means it will be n minus 1, and it will be 12. So this is kind of a handy way to get the number of trinucleotides and dinucleotides that can be obtained from a sequence. So I believe your assignments also could have these kind of questions in last week's in last week's section. I don't remember exactly, but okay, how do you apply this sort of calculation of dinucleotides or trinucleotides? You can apply those for, as I said, calculating a big lengths particular parameter. For example, it could be base stacking energy, hydrogen bonding energy, rigidity, flexibility, and so on. So in this particular question, it is asking the same thing. So here you have been given a particular sequence, that is ATAGAT, right? Here you have given this sequence, and alongside that you have been given the hydrogen bonding energy value for the small dinucleotide sequences, which are ATTA, AGGC, GA, CG, and so on, right? So these 6 constituent values for dinucleotide sequences you have given. So there is hydrogen bonding energy values, and then from that you have to calculate the ATA-GAT's total hydrogen bonding or average hydrogen bonding energy value. So anybody can mention what it could be? Just try to calculate. So try to first find out all of the dinucleotide sequences that you could have gotten from ATAG80, and then you just add the particular hydrogen bonding energy value for those dinucleotide sequences, and then you average it up. Okay, so that will give you the answer. Okay, so try to do it, and we'll just look into it. So just take a look at how many dinucleotides you can get, which dinucleotides you can get, and then you just add the values for all of those. You divide by the total number of dinucleotides. You should get an average help, right? Yeah, so I see some B answers in the chat. So again, as I said earlier, you start from each of the points, you move forward one step, you get a dinucleotide. So here AT, TA, then AG, GA, and then again AT, right? So these 5, I believe you are getting. Okay, so these will be ATPA, AGGA, and AT, and you have already available the hydrogen bonding energy values for individual components, right? So for example, for AT, you have here 7, right? So just add all of those up. So it will be -7, then -7, -9, -7, -7. Add all of them up, you will get -39. And since there are 5 constituent dinucleotides, you divide that by 5, and then you get a value of -7.80. Now, anybody can tell me why the energy values are negative here? What does the negative sign actually indicate?
Sir, for the spontaneous, it represents the spontaneous reaction that the reaction is environmentally feasible.
Yeah, that's probably one way to look at it also. So if you have
Or delta G is negative.
yeah, so Gibbs free energy, if you have it, so a negative value means that it is a stabilizing sort of reaction. You could say it in that way, right? So in the context of hydrogen bonding, so since two different components are coming together forming hydrogen bonds, so this -7.80 will mean that the total energy of the final, let's say, bonded structures together minus the initial energy. So the initial energy would have been when they were separate, right? They didn't have any bond formation. And what's the significance of this negative value? So does this mean anything in terms of favorability of the binding in terms of any energy? So if there is a value of, let's say, positive, or if there's a value of negative, so what does it usually indicate? Is it favorable or disfavorable for negatives or positives? It's thermodynamically stable, free energy value is negative. Yeah, so basically, when you have like a favorable kind of interactions, for example, a drug is interacting with a protein, or a protein is interacting with a protein by some sort of van der Waals interactions or hydrogen bonding interactions, you will usually see that the bonding energy for those are negative, right? So negative means that the final states energy, the final means the bound states energy. It could be covalently bound or hydrogen bond bound by hydrogen bond, or interacting by something like van der Waals, right? So the final interacting states total energy will be lesser than the initial states combined energies together. The initial state means when they were not interacting together. So since final is lesser, that means it's energetically lower, right? So if the system is becoming energetically lower, that means that process that they are following, that is particularly stable, right? System doesn't have to work to make the bonding. Yeah, so this kind of way you can kind of sort of calculate these parameters. So that was 7.80. I think we have gone through this question perhaps in the earlier classes. So we talked about different bonds here, for example, phosphodiester bonds between the adjacent nucleotides, right? We also talked about the glycosidic bonds, and we also talked about the hydrogen bonding in the other classes. So just again, very similar question. Could you again try to calculate this one? So it again asks for the hydrogen bonding energy for the sequence ACA, GCA, TG. Okay, it's a slightly lengthy sequence, and here you have been given the values for a lot of dinucleotide components, right? Dinucleotide sequence energy values. Just try to do it quickly and look at it. Okay, so same process, dinucleotides, and then uh, sum all of them up and then calculate the average. So ACA, GCA, TG. So that will have how many number of dinucleotides? We just start counting from here. So how many steps you can go forward one time, right? So the total number of constituting nucleotides minus one, that will give you the number of dinucleotides. And then you have to actually find out which dinucleotides you are dealing with here, right? So for example, here you have AC, you have CA, then you have AG, then you have GC, then you have CA, you have AT, you have TG. Okay. So hopefully the sound is still audible. Yeah. So what you do is that you add all of those values up, right? 7 point, yeah, it should be, I guess, -7 if you are counting in the sign as well. Yeah. So all of those added together, it will be -55. And if you divide by 7, it should be around -7.86, right? So these are some sort of basic kind of calculations that you might also face in your exam, these numeric type questions, right? So it's good to have some sort of practice of calculating this quickly. Okay.
Sir, do we have to remember the values for the exam?
No. You don't need to remember any of the values. The values will be given alongside the question. Okay. So you don't need to worry about remembering all of those values, any of the energy values, hydrogen bonding, base stacking, whatever. So mainly what you should basically, I guess, try to focus on are how to solve the numeric type of questions. And the particular things that you remember, it could be something related to some terminologies, for example, different terminologies for bioinformatics, databases, and so on, like schema queries, etc., etc. Let's see, if we're talking about proteins, then you might try to be familiar with uh, the characterization of different amino acids, right? The polar, the non-polar amino acids, the hydrogen bond forming amino acids, the sulfur containing amino acids, and so on. So those sort of questions, because those are kind of very, I guess, core concepts of the particular topics that are being covered. So those sort of concepts you might need to remember. You should remember, but here in this case, for example, the energy values you don't need to remember. You will be given all of the resources or values that you need for the calculations. Okay. So just a quick one here. Which of the following bases are for RNA or is for RNA? We know ATGC, right, for DNA, and then AUGC for RNA, right? So it should be uracil here. Okay. See one hand here. Yeah. You can unmute, or you can also ask questions in the chat. So these kind of like nucleotide structures, you should try to remember certain stuff about those. For example, the different bonds they are forming, right? What sort of nucleotides they are kind of composed of. Those sort of things you should try to remember. But otherwise, you will be given all of the necessary values that you need to perform any calculation on, right? So here in this particular question again, so it's not a dinucleotide or trinucleotide sequence based question, but it is asking for the content, the GC content from a given stretch of sequence, right? So the GC content calculation, you have to of course see how many G's, how many C's are there, and then the total length of the sequence, and then you can just divide the G's and C's by the total length, multiply by 100, you should get the answer, right? So anybody can tell what the answer should be? Yeah, so a lot of answer B. Yeah, so in this case, you find out just how many G's, how many C's you have. For example, we had 8, right? You divide that by the total length of the sequence, should be 17. You multiply by 100, you get 47.05%. So here the answer was B, right? Yeah. So you remember when we talked about nucleic acids or nucleotides, we had different bases, right? We had two kinds of them. We had purine and then we had pyrimidine, right? So anybody remember the structure, what they look like? So what was pyrimidines constituting of? Like what does pyrimidine constitute of? Is it aromatic ring 1, 2 aromatic rings, none, 3 aromatic rings? Yeah, so these kinds of questions are what I'm talking about. So this kind of sort of small scale structural level questions, compositional questions, these type of questions you might face. So try to remember those concepts. Okay. Yeah, so I see a lot of you have answered 1 aromatic ring, right? In this case, that was the correct one. So here, if you see, yeah, so in this case, you see that for purine, there are 2 rings, right? We have adenine and guanine as part of purines, right? And we have pyrimidines. For pyrimidines, we have cytosine, uracil, and also thymine. So these are 1 aromatic ring having bases. Okay, so pyrimidines in this case has have 1 aromatic ring. If it was asked for purine, then it would have been 2. Okay, that's fine. By the way, so the citations or the references that I'm giving here, so I had included those in the Google Drive folder. If you check carefully, so I've also included a couple of resources alongside the slides in the shared Google Drive folder. So there I have included certain PDF for books that I had also followed, and then certain research papers as well for coding, Python-based bioinformatics, and so on. So those kinds of resources I have included. So if you want to take a look into that particular subfolder, you can certainly do. Okay, I think it should be accessible to you all if you have the link to open that folder to the NPTEL platform. Okay. So now I guess we will shift the focus a little bit on to databases, which we had also learned in earlier weeks classes. By the way, so since we are talking about, we were just solving some problems regarding dinucleotides and trinucleotide sequences. So can anybody tell me why do you consider overlapping stretches of dinucleotides and trinucleotides? Why not non-overlapping stages of dinucleotides and trinucleotides? Any guess or any idea?
Sir, we can get an idea about the base stacking energy. So the bases are stacked on top of another, right? So in order to know the base stacking energy of these, so we have to know the hydrogen bond stacking energy. So they are calculated adjacent to each other, because if we skip a nucleotide, it won't be as much of a use.
Yeah, so yeah, that's kind of correct. So basically, if you consider non-overlapping stretches of dinucleotides or trinucleotides, you will miss a lot of local context, kind of, right? So, for example, I don't know. So, if in our earlier question, so for example, let's say, yeah. So here you had, for example, ACA, GC, ATG. If you considered only non-overlapping parts, so it would have been like AC, AG, CA, TG, and so on, right? And this is particularly a very small sort of sequence, right? A small length sequence. But if you had a really big kind of sequence, you would have seen that you in a lot of cases miss a lot of local context. By local context, I mean that you miss the importance of uh, certain nucleotides that are staying close by together, right? For example, you will miss a lot of small dinucleotides, let's say like GA, CG, or and so on, right? So if you had considered the overlapping sequences, so that means number one, your sample size would be a lot bigger, right? Your sample size being bigger, that means that you will get a probably more accurate data. Then you will get the local context. For example, if one cytosine is nearby one adenine, uh, then to get that, if you try to just get non-overlapping sequence, for example, AC and AG, then you will miss this particular effect of what happens if a C is just nearby A, right? So these sort of local parameters you will only get if you take or if you consider overlapping sequences, and it increases the sample size. So yeah, what you said was correct. So basically that's why we need to always consider overlapping sequence for dinucleotides and trinucleotides. Yeah, so database, as we talked about in the earlier session as well, Professor Pumiha has talked about. So these are basically a repository, electronic repository, where you store and organize data, and also you implement certain tools or programs that allows facilitation uh, or facilitates easy retrieval. Okay, so the retrieval has to be easy. That's what that is also a big feature of database, because if you are only storing the data and if you are not allowed to get any database on your particular complex queries and so on, so it would have not been that much effective, right? So here, alongside the high level of organization annotation, you need also the easiest retrieval possible for your particular search interest, right? So basically, a part of database that composes both these hardware and software parts, uh, and then it's basically the ultimate goal is to structure data into records. And for example, here you have multiple fields such as name, phone numbers, addresses, and so on. We'll talk about some of those in just a bit. And then the complex searches that you perform, so those are kind of called queries, right? So you can give query to a database. You can specify particularly what you want to get out of the database. Um, and if you talk about a lot of the characteristics for a database, so this will be the content, of course, the ontology. So that's the terms and definitions, the schema or the logical structure, the format of the data, the retrieval methods of the data. Then, as I said, the data will also have high level of annotation as well as links to relevant sources outside, so that you can go back to the different relevant publications or other databases or any other materials that is valuable, right? Or that the database itself has taken reference from. Now, if you talk about biological databases, so there are let's say three main types of databases, right? One is primary database, another is secondary database, and some databases are called specialized database. So primary databases, they mainly store original data, such as raw sequence structural data submitted by the community. So any idea about primary database or any example that you can give?
So EMBL is a primary database.
Yeah,
So GenBank also.
yeah. So EMBL, DDBJ, GenBank, those would have been your primary database. Should PDB be a primary database or secondary database?
It is a primary database.
Yeah, yeah. PDB is also primary database because it contains the crystallographic information directly from experiments, right? So yeah, GenBank, European Molecular Biology Laboratory, or your DDBJ, DNA Data Bank of Japan, PDB, etc. So these are your examples of primary database. And then when you process the primary kind of data or manually curate informations from the primary database and then add it to some other sort of database, so you create what is called secondary database, right? So they often have functional annotations. For example, here you have Swiss prot, protein information, resource, etc. Now, if you want to move, let's say, to a more sort of specialized database where it has information on specific niche kind of categories. So for example, if we have the sequence database for HIV or the ribosomal database project and so on. So these are again focusing more on narrow kind of research parts or narrow areas, right? So those will be kind of count as your specialized databases, right? Now, so just a quick question. We just talked about it. So if you want to get information from a database or a table or a combination of tables, so what do you perform or what is the request that is given by you called?
Query.
Yeah, so it's called a query, right? So that's one term. Uh, then there is another particular term relevant in case of databases. So that is called SQL. Any idea what it is?
It is a language. Actually, it's that structured query language used to write the query, how to present the query in
Yes.
computer format.
Yeah. Basically. So it's called structured query language. Yeah, I see hand also. You can ask the question or you can even give it in the chat. So SQL is basically, as you said, it's called structured query language. It is a language kind of used for creating and processing of databases. Okay, so it's particularly specially built for that purpose only. So the purpose of it is managing or manipulating what is called relational databases. Okay, so we'll just go through relational databases as well. So regarding the underlying database management system or DBMS, it is particularly a standardized way to interact. And then there are different types of SQL commands. For example, you have query commands, you have definition language, manipulation language, control language, etc. So for example, some common ones used, I guess, could be select to retrieve data for more tables, insert to add records, update for modification of existing table, deletes for removal, create for any new objects such as tables, indexes, and so on. Okay. Here I also have included when you get this particular, I guess I can just control and follow a link. It could be a kind of a helpful link, this particular website here. So apart from SQL or SQL, they also have different other sort of small scale tutorials for other programming language, for example, Python, Java, PHP, etc. So if you are interested in any sort of programming language or any particular language, you can get the basic information or some basic tutorials from this particular page. Okay, so here, for example, you have select star from customer. So star usually means all from a certain object or table, something like that, right? So here they have exercises and so on. Okay? So different commands, what they do, etc., etc. So you can try to go through this page also at your own time, and then you can see if you try to learn any sort of new language or any syntax or examples or commands or from those languages. Okay, yeah, so that's one thing. Now we just talked about something called SQL, which basically manages relational databases, right? So any idea what a relational database is in the context of the different database management systems? uh, so there are a couple of those. For example, SQL, Relational database is one of those, right? So there are others, for example, object-oriented databases, or hybrid databases also. C. So relational database is a particularly said correctly. So it is the data present in different tables that should have at least one common attribute, so that you can link those tables. So those tables should have some sort of common linkage, right? And that's how you get a relational database. So at least common attribute they should have, and it is the part of what we call a database management system, or it is one such category of DTMS. Okay. So here what it is saying is that the computer programs that have been developed to streamline organization, search, retrieval of data, those are called as database management systems, right? So that's called DBMS, or database management systems. And they do not only hold the raw records of the data, but they also have instructions or operational instructions that could uncover any underlying connections, right? And it is basically to facilitate efficient execution of searches, to manage different records, comprehensive search reports, and so on. And the type of categories that a database management system can be divided into, uh, those are relational database management system, as we talked about just now, and another one is object-oriented database management system. If you combine those, you can also get a hybrid sort of database management system. Okay, so when you have a relational database management system, the databases will be called as relational database. When you have object-oriented systems, they will be called object-oriented databases. And how basically the relational databases work is that they have multiple tables for organization of the data, and each table here, they are called a relation. Okay, they have multiple rows and columns, and then the columns there, they will represent individual fields, and then the rows, they will contain the specific values for those fields, and those will be particularly your records, right? Each cells or each records, right? And then there is also a shared characteristic which is called an attribute, and that will facilitate cross-referencing of the data present in other tables. Okay, now we have to give different queries to get the particular search results, and then you will get a unified report for quicker access to the information. Now, if you have a flat file database, relational databases will have comparatively high level of advantages from those as compared to those flat file databases, right? So the data retrieval will be very handy or very efficient as compared to the flat file databases. And you also have specialized languages like SQL or SQL to manage and to get information from the relational databases, right? So it is a very flexible way of organizing your data. Okay. So for example, here, if you have just a flat file database, right? So what this flat file database is particularly saying is that so you have the data for name, state, course number, and course name. So let's say this is some university's data sheet or database, and then you have the data for the students name, which state do they come from, what is the course that they are taking, so the course number, and as well as the course name, right? You see that the basically devising factor from the individual entries for the students, that is done by this delimiter or pipe right here. So you'll see this thing. Okay, so all of the entries are kind of divided by this via this, and for let's say each column, so it would be name, state, course number, or course name, you have uh, the values separated for commas, right? So for example, in this first entry, so John Smith would be the name, then Texas would be the state, right? Biol 689 would be the course number, and so on. So this is a flat file database. The problem with it is that it does not have any hidden information or a hidden instruction for facilitating a quick retrieval of the data. So if you have a complex query, for example, if you want to know, let's say, which students from the particular state of Illinois are taking the course, let's say biochemistry, right? PICH 441. Okay, so that's kind of a complex query. If you have to go through a large uh, volume of files to get that particular data, then it would have consumed a lot of computational resource, basically, right? So you have to manually go through every sort of lines and every entries of the data, uh, and then from there you have to get the particular answer to your big complex query, right? So that's not, maybe it's not a problem for any small scale databases, but if you have a large volume of data, like gigabytes or terabytes of data even, that becomes a big problem. It consumes a lot of resources, right? And a lot of time. So that's why you have relational databases where multiple tables are there, right? And then, for example, in table A, you have students 1, 2, 3, 4, 5. They name their states. You have student for table B. You have students as well as the courses. For table C, you have the courses as well as the course name. Okay. So you see that with table A and table B, there is one shared column that is called the student. Okay. You see with table B and table C, there is one shared column that is again called the course, right? So with table A and table B, it was the student, right? So now if you are asked from the particular city of Texas, which students are taking which course, so now you, in a way, you do not have to search through the entire database. So you do not have to go through all of the lines for a flat file database. You first search table A here because that contains the state information, right? So for example, the state of Texas, if you are searching for this particular state, you are seeing that, okay, for the state of Texas, searching through the entirety of the table A, you get student number 1 and 5 that are involved in this curriculum, right? Now, when you search for table B, you do not have to go through all of the students here. So you only go for student 1 and you only go for student 5, and you see which courses they are taking. So you take their course, basically the course code, and then you match the course code to the actual name of the course, right? So here you see only for table A, you had to go through the entire basically data set, the entire table here for table A only. For table B and then for table C, you did not have to go through the entire data set. Your search area was basically narrowed down, right? Just, yeah. So your search area was basically narrowed down here, and that is the benefit of having the shared attributes or the shared columns in a relational database is that your computational expense and your time for searching becomes a lot lesser, right? Alongside that, of course, these sort of databases, they will have instructions, they will have uh, basically programming languages such as MySQL to basically get the exact information you want through a programmatic method. So that's the advantage of relational database here. Here, as it is said, that by creating three different tables linked by common fields, the data can be easily accessed and reassembled. So that's another thing. So if you had to basically insert, let's say, certain entries in a flat file database, it would have been again kind of difficult, right? Match all of those up, like which particular entry field you should fill up, and then what would be corresponding to that in some other cell, and so on, right? In the case of relational database, since it's in kind of in a tabular fashion, so that also becomes a little bit more easier. Okay. So now there are certain limitations for relational database. For example, it says that it cannot handle hierarchical relationships between different items, and also sometimes it may not describe the data connections properly. Okay, so to kind of mitigate those factors, here you had the object-oriented database that has been introduced. So the object-oriented databases, they are particularly developed using different object-oriented programming languages, for example, C++. And here what they do is that they programmatically link different objects using something called pointers, and that sort of defines a relationship between them. Okay. So searching involves navigating through objects with the help of pointers linking different objects. So the object-oriented databases are also very flexible because it allows structured on a hierarchical relationships, complex relationships. And then if you have, for example, a hybrid approach, so that contains some of the attributes or some of the features of the relational databases, as well as some of the features of the object-oriented databases, which are also quite popular. Okay, because I think
Sir,
yeah, yeah.
sir, can you elaborate what kind of pointers do database use?
So if we just talk about in a, for example, visualizing manner, so here, for example, the database will have included something sort of this sort of box-like structures. Okay. So here, if you say the objects, this, for example, the every state was considered as an object. So here the first box was Kansas, Illinois, New York, Texas students, and so on, right? So then you have the students object which links to the state objects, particularly through something like this. So it is, you can think of it, it is like a sort of connection, like a programmatic connection between one box from the state object to another box in the student object, to another cell in the student object. Okay. And then the different student objects, they are also linked to the course object here. So maybe this visualization is of help. Here,
Yes, yes, thank you.
so it's basically kind of a programmatic link between the different objects or different box you can think of. One could be a state object, another could be a student object, another could be a course object, and so on. So in this particular case also, you have easy retrieval of data because the pointers are linking you directly towards where your search item that you want is available, right? For example, you have the student from the Texas that is linked to student number 5 that is linked to the math 172 course. So these sort of ways also the databases can be constructed. Okay. So here it says, for example, in the bottom notes, here it says that 3 objects are constructed and they are linked by pointers shown as arrows. Okay. So here also specific information finding that relies on navigating through the objects by the way of pointers. And that's how you basically get access to your particular query data. That's how you get the output of your particular complex query. Okay. I guess if you want to learn more details way about pointers, then you have to look into some basic concepts of some object-oriented programming language. Okay. For example, C or C++. Then there you will get the ideas about the pointers and so on. Okay.
Okay, sir.
How they are particularly constructed. Okay, so that was something regarding to databases. So I guess this particular question was in your earlier assignment, if I remember correctly. So anybody remember what the answer would be? Yeah. So the so as you say on the chat, so the videos, all of the videos that the TAs will take, so those will be uploaded to YouTube, and I believe you should have available one shared Excel sheet from the NPTEL platform for this particular course. So after each week's session, we'll upload the videos on YouTube, and then we'll get the we'll provide the link for the YouTube as well. We'll provide the link for the shared Google Drive folder where you can get the slides basically. Okay. So all of those links will be uploaded once we finish processing the recording and such. Then it will be uploaded to YouTube. Yeah. So here, as I asked, in drug design particularly, what's the role of computational biology? So any idea? So interestingly, if you talk about computational biology or bioinformatics as broadly also, so the factors such as toxicity, such as IC50 values, etc., while not directly, you can kind of get an idea of, right? So that's why in this particular case, I would say identify potential lead molecules. That's very correct answer, the most straightforward answer. Since here in this particular other formats, I had given measure kind of as the indicator for getting the value of toxicity or IC50. So for measuring something, of course, I have to perform the experimental procedures, right? However, a big part of computational biology also is not measuring but estimating different types of parameters. Okay, so for example, we had talked about earlier the ADMET ADMET estimators or the ADMET calculators, right? ADMET computing web servers. So ADMET is, of course, stands for absorption, distribution, metabolism, excretion, toxicity, right? And a lot of web servers are there where they have trained certain models based on already available experimental data, where they will link certain features derived from a small molecule to their physicochemical and pharmaceutical parameters, right? So that's how they will give you an estimate of the potential ADMET profiles of a particular new molecule, right? So in this particular case, you are not exactly measuring toxicity through the admit parameter scanning or admit searching. You are kind of giving an estimate of what the admit profile could be. So that's kind of the difference here. Similarly, I guess you could say that measuring IC50 is also, of course, not possible computationally, but you can get an estimate of the IC50 value if you convert, for example, something like binding energy to KD value, or in similar way, right? You can also, of course, train different models for predicting IC50 value and such also. Identifying drugs, that's sort of a very broad statement. So to exactly identify drugs, computational biology, while it is serving as a key part of that, it's not the entire thing. It's basically the initial part of a big pipeline where you also need to validate your computational data with experimental processes, experimental approaches, right? You have to find out in vitro, in vivo, as well as later in clinical trials or preclinical trials, etc., whether your drug is actually working or not. Okay, so that's how you ultimately get the drug approved in the end, right? So here the best answer would have been potentially identify potential lead molecules, right? So the lead molecules or potential lead molecules that could be identified via hit to lead screening, or something sort of like a molecular docking, or a virtual screening, or even if you talk about more computationally expensive processes, you can also try some sort of very robust free energy calculation methods. Okay, so those will give you an idea of uh, your binding affinity between particular target and the drug molecule. And then through computational biology, as I said, you can also uh, train your particular model for predicting admit parameters and so on, so that the drug will have any side effect or not. So while considering all of those factors, you can say, okay, this is our potential lead molecule that we can further proceed towards any experimental validation, or any in vivo validation or preclinical trials and so on. Okay. So here B would have been the answer. I see, yeah, on answer on hand in the chat. Okay. So you can also type in the chat as well. Yeah. So anybody knows what the most predominant amino acid in mammalian proteins are? So I believe this sort of knowledge-based questions you might face, particularly the types that Professor Bromihar talks about in his lectures. Okay, so just try to remember a few of those. It could be handy in your exam. Okay. Okay. So in this particular case, the L-alanine amino acid, that is the most dominant amino acid, or most predominantly observed amino acid in mammalian proteins. Okay. So here it is saying so the steric relationships of the stereoisomers of alanine L and D as compared, when comparison in comparison with the L and D glyceraldehyde. Okay. Uh, so for example, the R group of the amino acid, in this case the methyl group of the alanine, it is always below the alpha carbon, and L amino acids are those where the alpha amino group is on the left, and then the D amino acids are where the alpha amino group is on the right. Okay, so here you have the L-alanine, where the NH3 plus group is on the left, and here it is on the right, so it is a D-alanine. So the conformations you are kind of comparing directly with the L-glyceraldehyde as well as D-glyceraldehyde. Okay, that's how you get the D or L-alanine, and then the L-alanine is the particular one that is present most predominantly in mammalian proteins. Yeah. So, do you remember any of the amino acids that have R groups of aromatic nature? There are 20 standard amino acids. So, which of those had aromatic R groups? Anyone? So the amino acid classifications, their side chain kind of classifications, whether they are forming hydrogen bonds, whether they have any aromatic R groups or not, whether they are polar, whether they are hydrophobic. So these sort of particular set of parameters you might, I guess, need to keep in memory just a bit, right? Which could be handy. Yeah, please.
Sir, I have a doubt actually. So, when I was reviewing Michael sir's video, he listed proline in a hydrophilic residue.
Okay.
So I think so. But in Lehinger, as a standard biochemistry book, as I follow, it is a non-polar amino acid.
Yeah.
It is classified. Yeah. So
Uh,
what which case do I follow about proline?
yeah. So I believe the example that I had included also is also from Lellinger actually. Okay, so in this particular case, the tyrosine and tryptophan, they were the aromatic R containing amino acids. But here, as you said, in the classification of the amino acids, yeah, proline is non-polar, right? It's an aliphatic R containing group here in this particular case.
Yes, sir. He put it under hydrophilic as he put 10 in his video he said that 10 are hydrophobic residues and 10 are hydrophilic residues. So if we go by that, the proline doesn't count as a hydrophilic
Yeah,
if we go by Lehinger.
yeah, I think I'll have to check it actually what is said exactly here based on which context. But here, see, for example, what I took from Leninger is that proline is listed as a non-polar amino acid. And there is also another parameter that I guess we'll talk about in the next slide, uh, where it kind of measures the free energy change of a particular amino acid if you put into a polar solvent versus a non-polar solvent. Okay. So that free energy change will give us also the value of where this particular amino acid is more stable in which environment, which is also kind of linked to their hydrophobicity or hydrophilicity. In general, I say proline is hydrophobic or non-polar, but I have to check Professor Tomina's lecture exactly what is said there.
Sir, it's the lecture one from week 2.
Yeah. So as you see in this particular case, so there is a lot of non-polar R-containing groups as well as the aromatic R groups for phenylalanine, tyrosine, tryptophan. And then for the charged R groups, you will have or polar R groups, you will have serine, threonine, and cysteine as polar, and then you have asparagine and glutamine as kind of polar uncharged, or all of them. For the charged, you have positive or negatively charged. For negatively charged, you have aspartate, glutamate, and then for positively charged, you have lysine, arginine, histidine. So these kind of classifications you remember because in certain numerical problems as well as you saw earlier. They can ask you, for example, composition of positively charged amino acids or negatively charged amino acids, or polar uncharged amino acids in a particular stage of sequence, right? So it's better that you remember some of these categories here. Let's just see if I find that one. Yeah. So here also taken from Leninger, there was one particular value associated with all of the amino acids, which is called hydropathy index. Okay. As I said. So hydropathy index is basically a scale where you can see in which range the particular hydrophobicity or hydrophilicity of the R groups are basically situated at. Okay. Now, what does the hydropathy index indicate? It indicates the free energy of transfer of the amino acid side chain from a hydrophobic solvent to water. Okay, so if you have a amino acid and then you transfer it from a hydrophobic solvent to water, can anybody tell me in which particular case that transfer would be favorable? By which I mean that if the group is polar or if the group is non-polar. Anyone?
Sir, can you repeat the question?
I mean that if you transfer a amino acid from a hydrophobic solvent to water, will that transfer be favorable for a polar amino acid or a non-polar amino acid?
It's a polar amino acid.
Yeah. So because the polar amino acids they favor water, right? Or polar solvents. So that's why the transfer will be favorable for polar amino acids, right? So we know that when a transfer or when certain process or reaction is favorable, that has a delta G less than 0, right? So negative value, right? Negative value of delta G. And this particular delta G, since we are calling it hydropathic index here, so in this case, wherever you see sort of a negative value, that will mean that a transfer of that particular amino acid from a hydrophobic solvent to water, that is negative, right? So here you also see for groups such as, proline here is also having a minus 1.6 value. For example, very strong polar groups such as asparagine, glutamine, they have a very high negative value, for example, minus 3.5, minus 3.5 here. And then polar groups, for example, positively charged the groups minus 3.9, 3.2, minus 4.5, etc. So these are where the favorable transfer of the amino acid to water is seen, right? And then on the other hand, if the energy value is positive here, so a positive value for the hydropathy index will indicate that transfer from hydrophobic solvent to water is not favorable. So in that case, that will be classified as a hydrophobic side chain containing amino acids or hydrophobic amino acids, right? For example, strongly here you can see something like leucine, it has 3.8, right? And then here's one case where cysteine is classified as a polar amino acid despite having a positive hydropathy index because the SH or the sulfhydryl groups that is present here, that oftentimes acts as a weak acid and forms weak hydrogen bond with oxygen and nitrogen. Okay. So that's one way to also classify the amino acid. Yeah. So if you have, for example, you have been given certain amino acids. Here in this case, you have serine, aspartic acid, and threonine. Would you rank these particular ones with an increasing order of polarity? So can anybody give me the answer? So if you remember, there was a certain order or ranking of the particular functional groups for in terms of their polarity. So you kind of have to follow that sort of rank for the functional groups, and then you could get the answer. See a lot of answers here. So in this case, if you look at the structure of this particular amino acids, you have the C double O minus of the aspartate here. You have the CH2OH of the serine. You have the again you have another hydroxyl group here, but it is bound to a carbon at different position, right? And then here you have also a large number of or basically higher number of carbon chains. So in this particular case, if you see the polarity order of the functional amino acid groups, you will see that the carboxylic acid having amino acids that is staying quite at the highest kind of end, towards the highest end there. So here in this case, if you talk about the most polar particular amino acid among these, so the aspartic acid that would have been the highest value, right? Then you have serine, which has a OH, and then you have the threonine, which also has a OH, but it has a higher, uh, higher length of carbon chains. So if you see that the alkanes are kind of at the lower end, so combining all of that, you could say that threonine would be the least polar among these. Then serine would come at the midpoint, and then aspartic acid would be the last or the highest polar. Okay. So you can get, if you remember the polarity order of the functional groups in this particular way, in that case, you could also work out the different amino acids and their order of polarity. Okay. Anybody can say which of these following amino acids can form hydrogen bonds with their side chain, particularly the R group, the side chain R. So any idea? Uh, So hydrogen bonds are also a very important interactions that stabilizes the protein structures. First of all, of course, you know that your different secondary structure formation, for example, alpha helices, beta sheets, they have a particular sort of hydrogen bond pattern, right? Hydrogen bonding patterns. Uh, so alongside stabilizing of the secondary structures or even stabilizing of the 3D fold in certain cases, right? The hydrogen bonds also play an important role in uh, interacting with multiple components, you can say. So if you have a protein, if you have a drug molecule or a ligand, so those can form strong hydrogen bonds. Very high level of interaction based on hydrogen bonds only. That's possible. If it is protein or peptide, that is also possible that they have high degree of hydrogen bond formation. We should lead them towards a favorable binding energy. Uh, and then, of course, you know that for nucleic acids also, hydrogen bonding are very critical for their overall structure, right? So in this particular case, we are talking about the amino acids, and here it would have been serine. Okay, so serine has a side chain OH, and as you know, the side chain OH that can be used for forming hydrogen bond with different particular uh, elements, for example, right? So since serine has a side chain OH, then it could be forming hydrogen bonds. So these are also again some sort of knowledge-based questions that Professor Kumi had talked about. So mainly for the channel proteins, so they have both of these properties or features. Okay, so there is gating features, and there is also selective conduction of ions, and that's the main particular defining factor for the channel proteins. Okay, so these sort of questions also Professor Kumi had talked about. So you should try to remember these. Let's see if we can go to the sequence alignment parts on what. Okay, so this is kind of a sort of an analytical type of question. Can anybody tell which of the following is not true for these particular question? So the first option says that the hydrophobic or hydrophilic character of the amino acids is important to the secondary structure. The second option is saying that the intramolecular hydrogen bonds are important to the secondary structure. Then also the alpha helix, beta beta sheets, etc. They are the examples of secondary structure. Yeah. So in this case, the hydrophobic or hydrophilic character of amino acids, that is not quite as important to the secondary structures. It is of course very important for the overall fold of the protein, because the hydrophobic part will stay at the core of the protein away from the solvent, right? The hydrophilic amino acid portions, they will stay relevant or exposed to the particular solvent. So the protein will fold in a way which minimizes the interaction of water with the hydrophobic amino acids and maximizes the same with the hydrophilic amino acids in a sense, right? So the hydrophobic or hydrophilic characteristic is important for the tertiary or 3D fold of a protein, not as such the secondary structure, right? The secondary structures are stabilized via hydrogen bonds. So yeah, so these kinds of questions you can also get. Yeah, this is kind of an example of secondary structure formation here. So particular sort of patterns, right? And for evaluation of these secondary structures, you also know you have something called. So what plot is this on the bottom right?
Ramachandran plot.
Yes, so Ramachandran plot. There we have the particular phi and psi angle based representation, and there is certain portions you will see basically in at certain phi psi ranges, particular sort of secondary structures they reside at. For example, the right-handed alpha helix will be in this particular position, in this particular dot position here, right? So parallel beta sheets, anti-parallel beta sheets, left-handed alpha helix, and so on. You will get different sets of ranges, right? So you see in a lot of cases, mostly when you try to model, let's say, a 3D structure of a protein from a sequence, right? Uh, then after you get the initial model, you will also have to calculate the Ramachandran plot favorability, right? So if the Ramachandran plot is showing that most of the amino acids is of course Rama favored, so then that's fine, of course, but it is not up to the mark, then you have to consider again certain parameters or start remodeling or try to optimize the structure in a way, right? You might try to energy minimize the structure and then again try to calculate the Ramachandran plots and so on to see whether basically most of the residues are favored in this case. Okay, so that's one thing. And then yeah, you have particularly the proper or ideal values for the phi psi angles for different types of structures. So you don't need to realize this. It's just for information. Okay, you can get the exact idea from the Leninger book, of course, but there are a lot of web servers that will do the calculations for you if you're doing a large scale calculation on certain proteins and so on. So you don't particularly need to remember everything from this range value, but it's good to get an idea of. Okay, so here's another question. So we talk about the hydrophobic clumping, right, or hydrophobic clustering. It's basically when you drop, let's say, or when you give a long stretch of sequence of hydrophobic residues in water, right, or in a polar solvent. So what will happen is that they will clump together. They will try to cluster together or clump together, right, in an aqueous solution, for example. So any idea what is the driving force for this particular occurrence? Why do hydrophobic amino acids tend to cluster together in an aqueous solution? Do they try to form covalent bonds? Is there any electrostatic interaction that is possible? Does it have to do with something like entropy, or is there any hydrogen bonding? So I see a lot of you have said C, right? So yeah, in this particular case, a favorable increase in entropy would have been the answer. So what is entropy? Anybody can mention? What does entropy indicate, and why, if you say hydrophobic clustering might be associated with entropy?
Sir, it is a measure of disorder or randomness.
Yes, it is a measure of the randomness or the disorder in a system, right? So the isolated systems, as you know from the second thermodynamics law, it will have a tendency towards an increase of entropy, right? An increase in randomness. Okay. Now, what happens when you drop certain hydrophobic residues? So let's try to see that. So let's say here you have a bucket of water or something. Then you drop different hydrophobic residues together. Okay, so they will tend to clump together, right? But firstly, what happens is that uh, because you are dropping the residues onto the water molecules surrounding those, they actually form something like a solvation layer or a solvation shell around the hydrophobic amino acid residues. Okay, so these solvation shells, they are actually highly stable kind of structure because the water molecule that is forming hydrogen bonds together and then surrounding this particular hydrophobic amino acid residue, that structure is very highly ordered. Okay, so that is why that particular structure in this case it sort of has a low entropy, you can say, right? Now, if you imagine another situation where these hydrophobic amino acids will be clustered together, right? Then of course there still will be a solvation shell around this particular hydrophobic amino acids. But if we compare the surface that or the volume that this particular solution shell will occupy, you will know that it is less than what was earlier when the hydrophobic amino acids were separated together. So here, if you add all of these solution shells, let's say surface area together, and then you get a value, and then you do the calculation finally for whenever these all of the amino acids have been clustered together. So if you perform the surface area calculation for this entire solution shell, this big particular solution shell, you will see that the sum of all of these individual small ones that will still be bigger than the ultimate final big one when they are clustered together. Okay, so the surface area or volume covered by this particular solution shell, that is decreasing. Now, since the solution shells, they represent ordered structure, so that means that uh, they represent more kind of stability, right? Not randomness or not disorderedness, right? So since the surface area is decreasing, so that means in this case, the amount of the orderedness, you can say, is decreasing. Vice versa, the randomness is increasing, if you think of it that way. And that is why the entropy will increase in this case, right? So in this particular case, a favorable increase in entropy you can see. So to mitigate, or to slightly reduce the solution shell or the water shell that is formed around the hydrophobic amino acid residue, to slightly reduce its overall surface area covered, so that the entropy is increasing. So that's kind of one way to look at this particular hydrophobic clustering. Okay. So again, some knowledge-based questions. For example, Professor Kumiha also talked about this. So mutation of GLU6 to valine, so glutamic acid 6 to valine in hemoglobin, that results in sickle cell anemia. Okay. So if he talks about any of these questions, do remember those, because that will be relevant in the exam situations. So which of the amino acids have imino group? You remember? What is an imino group, and which amino acid could have an imino group as their side chain? So imino group is particularly if you have all of the structures here. So this particular group for proline, that is called an imino group. Okay. So the proline is the one having a side chain imino group. Okay. Again, which amino acid cannot rotate a plane polarized light? If a question is asked in this way, anybody can answer. Plane polarized light. Which amino acid cannot rotate? So that basically talks about your optical activity, right? Actually, if you look at all of these answers here, none of those was correct actually.
Yeah, yes, sir. Actually, I was thinking.
Yeah, yeah, basically. So in all of these cases, you see different groups are attached. Yeah, yeah, different groups are attached to the particular central carbon. So all of them are able to rotate plate polarized light. All of them are optically active. Only glycine, you do not have the side chain, the R group, right? Instead, you have a hydrogen there. So since two hydrogens are attached to the central carbon, so that means that that one is not able to rotate plate polarized light. Okay. So these type of questions you can get. That's fine. Again, the proteins classifications, for example, what could be the structural proteins, what could be the enzymatic proteins. For example, here structural proteins are mainly fibrous proteins in nature, right? The globular proteins are your enzymes and so on. Okay, now you talk about or you start to talk about something called sequence alignment, right? So sequence alignment is basically when you align uh, all of the, you try to find basically perfect alignment between two or multiple sequences. And of course, based on the alignments, you can see whether, let's say, two sequences are at a close or further evolutionary distance. If a certain region of the sequences are aligning together, so there could be some functional relevance to that short stage of sequence and so on and so forth, right? So sequence alignment is a big part of bioinformatics. And to understand sequence alignment, you also have to, get an understanding about, of course, different processes that the sequence alignments can be performed to, or something called indels in this particular case. So oftentimes you will see that for proper alignment, or whatever is giving you the maximum alignment, it does not align each and every single component of the sequence together. So in certain cases, you will see that there are some gaps present in between. So it could always happen that the gap version is kind of giving you a better alignment score. So that gap is called indel in a lot of cases, and indel comes from insertions or deletions. Okay, so insertions and deletions together can give you an indel or a gap. And since in this case we are not concerned about whether a particular amino acid was removed from sequence 1 to get sequence 2, or a particular amino acid was added from sequence 2 to get sequence 1. So we are not talking about the chronology of the process that happened here. Both insertions and deletions are counted together. We are only interested in the gap. So that's why any gap when you have, you talk about indels, both of them together. Okay. Now, the basic method of comparing sequences via sequence alignment that Professor Komiha had mentioned, or Professor Komiha had talked about, that is the dot plots. Okay. So the dot plots are basically where you have, for example, two sequence, and then you write the individual components of one sequence on the top. You write the second sequence on the left hand side, and then you try to place a dot wherever you see a match. Okay. Uh, and if you get, for example, a diagonal kind of match on that particular region, you will see that the sequence, particular components of the sequences are matching together. So that is a way to give you kind of a basic sort of alignment. It's called a dot plot, by using a dot plot. So here you have, for example, at x-axis and y-axis, you have two sequences, right? So if you have these two sequences on the bottom reader on the left hand side, ADERFE in this particular fashion, let's say, and then if you try to place the dots, so usually in some cases you will also have the x-axis sequence at the top, but let's say here we are calculating in this way. Okay, so we have the first sequence at the bottom, we have the second sequence at the left, and then we are placing dots exactly at the matching points. So here we are taking what you call exact kind of matches, right? We are not using any windows, or we are not concerned about any stringency and so on. So could you probably try to figure it out where a continuous dots of more than 3 are observed? Is it above the diagonal? Is it below the diagonal? So in this case, the diagonal will be the main diagonal of this box. Okay. just place the dots on the particular matching sections, and then you can see. So I see a lot of you are seeing A. Okay, so how do you match the dots? You match the dots first by going through every element, for example, for the x-axis sequence, and then you see whether there is any match on the y-axis sequence for that or not. For example, you try to tackle L here, right? On the y-axis, you do not see any L, right? There is A, D, E, R, F, E. So you just leave this one. Okay, so after that, you talk about E. And then on the y-axis, if you see, there is one E here. Okay, so these two E are matching. So then you put a dot here. So that is one dot that you have put in the plot. Another E you can see here. So then you can put a dot here. Okay. If you put these two dots, so those are occupied, then there is A, right? So for A, you see one match here. So one dot should be here, right? At the bottom here. And then there is nothing else. So for D, you see one dot. And then for E, you see also one dot. And then for, sorry, two dots. And then for R, you also see one dot. Okay. Now, as I said, here we only consider the main diagonal. So since we are particularly keeping the x-axis sequence at the bottom, so here I was talking about this particular diagonal. Okay, so that is the kind of the main diagonal of the box here, and then here you see that the diagonal that we have found for continuous dots of more than 3, so the 4 dots diagonal, that is kind of below the main stretch of diagonal for the main box, right? So that would have been below the diagonal. Now, an interesting thing here is that why we are talking about the continuous stretches of dots in a diagonal position. So because the diagonal position means that both on the x-axis and both on the y-axis, if you just go for the corresponding element of that dot, you will find a match, right? So that means whenever there is a continuous match, we can see that it is matching both on the whenever there is a continuous stretch of dot at a diagonal in a diagonal fashion, you will see that the x-axis element as well as the y-axis element, these are matching together. Okay, so these ADER and then this ADER, that is matching together. So if you want to find out an element, then the dot plot is a kind of a basic good method, because now you know how to align this particular set of sequences, right? So the alignment would be kind of like this. If you have LEADER, so you know that the ADER are aligning together. So for the second sequence, your alignment will be something like this, for example, right? ADER and then FE. Okay. So here you see that the ADER part is common. On the left, on the sequence 1, you have LE on the left. For the sequence 2, you have FE on the right. But you have a common part. You have found kind of a common domain or a common motif for to base your alignment off, right? So that's why we are talking about the continuous stretches of dots with a diagonal fashion, and it can give you an idea about the alignments, right? Now, it's a basic sequence comparison techniques. So as I see in the chat, where do you use dot plots? So dot plots can give you particularly basic idea of the alignment of, for example, large scale sequence, two large sequences, let's say, right? And it can be useful in certain contexts by finding an stretch of diagonal present dots. You will be able to identify which stage of the sequences are matching. So there are two ways that could be useful. One is that you are actually trying to match one sequence with another sequence, and then another way is that if you try to match the same sequence with the same sequence, then wherever you are seeing continuous stretches of dots, you will know that there are repeated patterns of motifs. Okay, the motifs are present repeatedly. So that's also another way you could use the dot plots in a basic way. Okay, yeah, it's kind of an alignment algorithm, you could say. Okay. So here the matrix alignment the dot matrix based alignment technique is explained. So basically sequence A and B, they are represented on a matrix. Matrix basically means that on x-axis you have something, on y-axis you have something. Then you have cells corresponding to those, and then you try to present the matching cases in the terms of dots, or in the form of a dot. Okay, so the matrix is filled with dots indicating matches between the characters in sequences A and B. Okay, it's working. And then starting with the first character in B, each character is compared with the entire sequence, which we just did earlier. So starting with the very first of the x-axis sequence, we compare that element with every element of the other sequence, right? And then wherever you found a match, you just put a dot there. Okay, for all of the elements in the second sequence or the first sequence in any case, you have to repeat this process to fill out all of the possible points that could match. And then if you identify a diagonal rows of dots on the matrix, so that will be where your similar sequences are present. So that will give you an idea about, okay, this part could align with that part, right? Then if you have a lot of isolated dots, for example, in this right-hand side plot, you see a lot of dots, right? It is filled with dots, but all of these dots do not represent a pattern. Okay, so where you see a pattern is where something like this is present, where a diagonal stretch of dot can be visualized on the plot. Okay, so that is the aligning part. Other dots are mostly random, right? So it is basically for a good visual representation of sequence similarities and also differences between sequence A and 2. So you can find out similarities, you can compare the differences as well. So let's say here we are just trying to align these two particular sets of sequence. So here again we are taking just as the previous one. So we'll try on the left hand side the first sequence, on the upper hand side second sequence. We'll just place a box here, and then we'll try to put all of the dots in between, right? So for example, here you have A versus A dot, right? Then A versus A, another dot will be here, I believe. So the page is not working. Yeah, here. Then let's see, you have C versus C. So C versus C, this could be here, this could be here, right? So on and so forth, right? So if you fill out this entire box with dot, you get something like this. Now here in this particular case, we have used a window of 1 and a stringency of 1. Any idea what this window and stringency could tell us about? What is the usage of window or stringency in this particular case? So number of connections allowed. Number of connections allowed, yeah. Number of connections allowed. So it's kind of, if we talk about exactly what it is that sometimes when constructing a dot plot or when visualizing sequences via dot plot, what could happen is that if you have a very large set of sequence or a large length of sequence and you want to match it with another large length of sequence, right? So the plot that you are getting, that could be basically overfilled with dots, right? It could be very kind of clumsy, cluttered, and not all of the patterns that could be possibly present there will be visible here because it is so cluttered. It's filled with dots everywhere, right? So that particular dot plot, when we talk about window 1 and stringency 1, it means that for every element of each sequence, we are trying to put a dot here. Now, to remove the clutterness or remove the number of dots that are present here, what we could do is we could do a sliding window sort of approach where we do not fill each and every element matching, but based on a small window of a certain stage of sequence. For example, let's say we take a window size of 3. So we will not match every single element to another single element of the two sequences, but we will match a 3 set a nucleotide sequence of the first one with the 3 set nucleotide sequence of the second one. And then based on how stringent you want your matching to be, for example, does all 3 elements need to be matched with all 3 elements there? Does 2 elements need to be matched in positioning with 2 elements of the other? Based on that, you will put a dot. So if I just show you the example here, so window 1 stringency 1, that is basically every dot at every match point. So at every element match, you are getting, right? You are getting 1 dot. Now, if you had a window of 3 and a stringency of 2, what will happen is that you will first consider these two, let's say. Okay, so here you have considered not one, but you have considered 3 elements of this first sequence together. So ACA together, and then ACG of the second sequence together, 3 elements, right? Now, if you have a stringency of 2, so that means that out of this 3 element window, 2 elements need to match exactly in positioning, right? And only then you will put a dot at the midpoint or at the intersection point. So here the intersection point would have been this, right? In this point. So based on a stringency of 2, a stringency of value 2, should we put a dot here or not? Any idea? Any guess? So we just talked about that we are trying to match not one element versus one element. We are trying to match 3 element versus 3 element of the 2 sequences. So the first sliding window that you are getting, it's ACA for this particular sequence, and then it's ACG for this particular sequence. And for stringency, we are saying that among all of these 3 elements, does exactly 2 sequence match in terms of their positioning? So if they match, then we'll put a dot here. Any idea? So it's more than 2. So yeah, more than 2 is fine. So stringency is basically, if it is more than 2 in some case, the stringency is basically meaning that at least 2 of them have to match together. Okay. It's basically the lower limit or the cutoff of how rigid you want your matching to be. Okay, so here in this case, for example, you have ACA versus ACG, and as you see, the first 2 elements of each of those are A and C, right? So here you have AC, and then here also you have AC, right? So that means that the 2 elements, the first 2 elements of these 2 window are matching exactly, right, in exact position. That means considering a stringency of 2, we can put a dot here. Now, let's say for the next step, now we are not considering ACA versus ACG, but we are just moving the window a little bit further. Okay, so you will calculate again in terms of overlapping sliding windows. But for example, let's say in this case, it's CAC versus CGC, right? So you see the first element of both of these 3-stage sequence is C. The third element is also C. So that means with a stringency of 2, we can match, we can put a match here, right? And match meaning we can put a dot at that intersection. Okay. So with a stringency of 2, they are matching. So we had 1 dot there, and then now we have an extra dot 2. So can anybody tell me, for example, if CAC we were to match with ACC, or in this particular case, in this window ACG. So if you are trying to match, let's say CGC with CAC with ACG, okay. So will there be a dot at that intersection with a stringency of 2?
No.
Yeah, no, because none of the elements are matching, right? So for stringency of 2, you need to have exactly at least 2 elements matching positionally. Here it's not that case, right? So anyway, so we had put 2 dots here, and then let's say another step here would be ACG versus ACG. So of course ACG, ACG, all of the elements are matching. So now you have another dot here, and then let's say if you go further a step, here you have ACG versus GCG. So the second and third elements are matching, right? So ultimately you put a dot here also. Now, what is the final outcome of putting this stringency and the window size here? You will see that the number of dots which you initially had in your window size 1 and stringency 1 calculation, if you compare that with your final result of putting a window size of 3 and stringency of 2, you will see that there are much lesser number of dots, right? Much lesser number of dots in the table or in the matrix. Here in this particular case, we might not get an exact idea of whether this was any beneficial to us or not. But in the case where there is a large length of sequence analysis is being performed, in that case, you might see in some cases introducing certain window size and certain stringency limits that will make us easily identify the patterns that are present in the sequences. Okay. For example, here you have, let's say, two examples. On the very first figure 3.5, it is the dot matrix of amino acid sequence of lambda ci, and then you have the dot matrix of DNA sequence, right? So these are kind of the examples of the dot matrix here. Okay. So I have also included a particular link in this slide. I can go to it directly. Yeah. So in this particular website, so it's made by Barry Grant, and they have a research group where they work with a different type of software suites. They develop basically different types of softwares that are useful for data analysis and such in terms of in biological contexts. For example, protein-related data, MD trajectory related data, and so on and so forth. Right. So they have made this particular just visualization tool here. So here, for example, you can do is they have given a protein dot plot along with a DNA dot plot. Okay. So if you just increase the window size and stringency, and you can here they have also implemented a moving window step size. So now you are not considering every overlapping window, but you are ultimately limiting that by how many steps a window will move forward to get the dot plot representation. Right? You can try to play around with this sort of window size, stringency size, and then match stringency, window step size, and so on. And then you will see that if you increase these parameters, sometimes you will see that the number of dots are very less, right? Becoming very less if you increase the window size and so on and so forth. Okay. For example, with a window size of 4 and then moving window step of 2 and then stringency match of 2, you are getting this kind of dot plot, right? If it had been the original dot plot where no value was included, it's the exact original step or the exact original match to match calculation, every point to point calculation, you will see that the dot plot is now more cluttered. Okay. So that's basically the idea here. The example, there are other examples also of how you can utilize the dot plots in pattern finding in the sequences, but that's kind of the basic of it. Okay. Yeah. So I think today we'll just wrap up the class, wrap up the session, and then on the next Monday we will discuss the further kind of concepts, the other concepts related to dot plot and so on, and then also the next set of alignment concepts, right? For example, different alignment algorithms that will be available on the course as well, instructed by Professor Krumiha. So please do check out the lectures carefully, of course, because the concepts that are covered there, they will also be available for your exams, and those are very foundational kind of core concepts of bioinformatics. And yeah, I guess we again meet on the next class then. Okay.
Thank you, sir.
Yeah. So thank you everyone again for joining the course. Also try to join the other TA session that will also go live on Tuesday, right? I guess at the same time, right, 6 to 8, I think we have the time slot for. And then all of the sessions will be available for the available for later viewing in YouTube also. So the links will be shared. So for proteomics, okay. So I'll try to search any book, any readily available book for proteomics, and then I'll probably include it in the folder, okay, in the subfolder that I had created. Since it's the last day of enrollment, can you suggest a good ML course on SPIAM? Okay, I particularly do not know of a ML course. So is it that for this particular course, it is the last, the last day of joining, or is it for every course that's there? So that's a course duration. Okay, so it's the good one here. I see exam date, enrollment 26/1. Just a moment. I'll try to log into this course and I'll see. I am able to join. So this particular course I'm seeing, it's the Introduction to Machine Learning. It's provided by Professor Baladaman Ravindran from IIT Madras. Since it's kind of an introductory course to machine learning, as it is said, so I think this one would be good. But here it also said the enrollment was up to 26th January 2026. So I don't know if it's available to join yet. Just a moment, I'll see. Introduction to machine learning course. Yeah, I don't see any joining date here. It only says learners enroll. So I think the 26th January was the last date for this one. I will see other posts available. There is one from IIT Kharagpur as well. The earlier year's iteration. It's not showing the current year. Engineering science. So a lot of these courses that I'm seeing, the enrollment actually ended on 26th January. So I think you have to go through the list and then see if you are able to join in any of those. Okay, because I'm not currently finding. Yeah. I'd say that for if you are particularly interested in NPTEL courses, just uh, try to see whether they are taking any learners for a particular machine learning course or not. Because two courses I saw, they had 26th January as last date of enrollment. Our bioinformatics courses last date is today, so it's possible that some other, courses will also offer this kind of extended duration for the deadlines or the joining deadline. But if you can't find any NPTEL course right now, again, you can look through other resources for getting your knowledge based on machine learning. You can also come back in the next iteration of this course, because I believe that this course it will again be available for new learners in the next year as well, right? So you can try to learn there also, similarly to the other courses. But right now, I'd say that try to go through a lot of let's say resources freely available on YouTube to get your fundamental ideas on machine learning. I would say the best way to try to learn is to try to do a hands-on implementation side by side, for example, right? So if you are working in any Python environments and so on, so you should get or try to look into a course where they are also providing kind of hands-on tutorial as such for analyzing data sets, data analysis like the basic libraries, firstly, for example, pandas, etc. Right, pandas, numpy, matplotlib, then ultimately moving to a scikit-learn and then how they're implementing machine learning and so on. So alongside learning the theories, of course, you need to particularly try out the hands-on so that the practical application is also uh, you get familiar with, basically. Okay. So try to do it that way, and then if NPTEL is offering a course in the next semester, you can try to look into it as well. Or if there are any other courses for machine learning available, then you can try to look into it as well. So the ones that I looked into are from IIT Madras and IIT Kharagpur, but those are not allowing joining right now. Let's see. Okay. Yeah. Okay. So again, thank you so much all of you for joining. And then again, we will meet at the next week's class. Okay. Thank you.